Dear students, Assalamu alaikum. The objectives of today's lecture are to learn about some of disorders of urinary concentrating ability, and secondly about the control of extracellular fluid osmolarity and sodium concentration. Disorders of urinary concentrating ability. Impairment in the ability of the kidneys to concentrate or dilute the urine can occur due to number 1, inappropriate ADH secretion, impairment of countercurrent mechanism, or inability of the distal tubule, collecting tubule, and collecting ducts to respond to ADH. The first of these disorders is the failure to produce antidiuretic hormone. And this failure to produce ADH may be due to central diabetes insipidus, which is caused by head injuries, infections, or maybe due to congenital causes. In central diabetes insipidus, the distal tubular segments cannot reabsorb water in the absence of ADH. This results in the formation of large volume of dilute urine. As a consequence, the thirst mechanisms are activated. So, as long as the person drinks enough water, large decreases in body fluid water do not occur. If water intake is restricted, then severe dehydration can rapidly occur. The treatment of central diabetes insipidus is by using desmopressin, which is a synthetic analog of antidiuretic hormone. It acts selectively on V2 receptors to increase water permeability in the late distal and collecting tubules. Desmopressin can be given by injection as a nasal spray or orally. Under certain conditions, normal or even increased levels of antidiuretic hormone are present, but the renal tubules cannot respond to ADH. This inability of kidneys to respond to ADH is referred to as the nephrogenic diabetes insipidus. Nephrogenic diabetes insipidus can be due to either failure of the countercurrent mechanism or failure of distal and collecting tubules and collecting ducts to respond to ADH. Large volumes of dilute urine are formed, which causes dehydration. Drugs affecting function of loop of Henle. Diuretics. Furosemide can compromise urine concentrating ability. Similarly, drugs affecting functions of distal nephron segments, including lithium and tetracyclines. Nephrogenic diabetes insipidus can be distinguished from central diabetes insipidus by administration of desmopressin. Lack of decrease in urine volume and an increase in urine osmolarity within 2 hours after desmopressin strongly suggests nephrogenic diabetes insipidus. The hypernatremia can be managed by low sodium diet or by administration of a diuretic to increase sodium excretion. Coming to the second topic of today's lecture, control of ECF osmolarity and sodium concentration. Regulation of extracellular fluid osmolarity and sodium concentration are closely linked because sodium is the most abundant ion in the extracellular compartment. Therefore, plasma osmolarity, denoted as POSM, can be roughly estimated from the plasma sodium concentration P sodium as equal to 2.1 multiplied by plasma sodium concentration. Two primary systems regulate the concentration of sodium and osmolarity of extracellular fluid. Number one, Osmoreceptor-ADH system, and number two, thirst mechanism. Let's first study the Osmoreceptor-ADH feedback system. An increase in extracellular fluid osmolarity causes the osmoreceptor cells located in the anterior hypothalamus to shrink. Shrinkage of the osmoreceptor cells sends nerve signals to supraoptic nuclei, which relay signals to the posterior pituitary gland. These action potentials or nerve signals to the posterior pituitary stimulate the release of antidiuretic hormone. Antidiuretic hormone enters the bloodstream and is transported to the kidneys, where it increases the water permeability of the late distal tubules, cortical collecting tubules, and medullary collecting ducts. The increased water permeability in the distal nephron segments causes increased water reabsorption. Therefore, water is conserved in the body. This causes dilution of the solutes in the extracellular fluid, leading to a decrease in extracellular fluid osmolarity. The opposite sequence of events occur when the extracellular fluid becomes too dilute, that is, osmolarity is decreased. This figure shows the osmoreceptor antidiuretic hormone feedback mechanism for regulating extracellular fluid osmolarity in response to a water deficit. Control of ADH secretion. This figure shows the neuroanatomy of the hypothalamus, where antidiuretic hormone is synthesized, and the posterior pituitary gland, where antidiuretic hormone is released. Antidiuretic hormone secretion is increased by an increase in plasma osmolarity, decreased blood volume, decreased blood pressure, nausea, hypoxia, and by drugs including morphine and nicotine. So all these factors increase the ADH secretion. On the other hand, ADH secretion is decreased by a decrease in plasma osmolarity, increased blood volume, increased blood pressure, and by drugs including alcohol, clonidine, haloperidol. The thirst mechanism is the second of the primary systems that regulate the concentration of sodium and osmolarity of extracellular fluid. Thirst is defined as the conscious desire for water. Although kidneys minimize fluid loss, adequate fluid intake is regulated mainly by the thirst mechanism. The nervous system centers for thirst are present in an area located anterolaterally in the preoptic nucleus of the hypothalamus. The neurons of the thirst center function as osmoreceptors to activate the thirst mechanism. Increased osmolarity of the cerebrospinal fluid in the third ventricle has essentially the same effect to promote drinking. The organum vasculosum is likely to be involved in mediating this response. Now let's study the stimuli for thirst. One of the most important stimulus is increased extracellular fluid osmolarity, which causes intracellular dehydration in the thirst centers, stimulating the sensation of thirst. It helps to dilute extracellular fluids and returns osmolarity towards normal. The second stimulus for thirst is decreased extracellular fluid volume and arterial pressure. This stimulates thirst by an independent pathway. There might be no change in plasma osmolarity, and this stimulation occurs due to neural inputs coming from arterial baroreceptors in the circulation. The third important stimulus for thirst is angiotensin 2. It acts on the subfornical organ and on the organum vasculosum. Its effect on thirst helps to restore blood volume and blood pressure towards normal, along with other actions of angiotensin 2 on the kidneys to decrease fluid excretion. Another stimulus for thirst is dry mouth. A thirsty person receives relief from thirst immediately after drinking water, even though the water has not been absorbed from the gastrointestinal tract. Gastrointestinal stimuli like gastrointestinal distension partially alleviates thirst. However, relief of thirst sensations is short-lived through GIT or pharyngeal mechanisms. The desire to drink is completely satisfied only when plasma osmolarity and plasma volume returns to normal. Another important point about the thirst mechanism is the threshold for drinking. When the sodium concentration increases only about 2 milliequivalent per liter above normal, the thirst mechanism is activated, causing a desire to drink water. This is called the threshold for drinking. Coming to the role of angiotensin 2 and aldosterone, although these hormones, angiotensin 2 and aldosterone, they increase the amount of sodium in extracellular fluid, they also increase the extracellular fluid volume by increasing reabsorption of water along with the sodium. Angiotensin 2 and aldosterone regulate sodium excretion by the kidneys, but remember, they do not regulate extracellular fluid sodium concentration. I repeat, they do not regulate extracellular fluid sodium concentration. Therefore, angiotensin 2 and aldosterone have little effect on sodium concentration except under extreme conditions. So, there are two primary reasons why changes in angiotensin 2 and aldosterone do not have a major effect on the plasma sodium concentration. Firstly, angiotensin 2 and aldosterone increase sodium and water reabsorption by the renal tubules, leading to increases in ECF volume and sodium quantity, but little change in net sodium concentration. Little change in net sodium concentration. And secondly, as long as the ADH thirst mechanism is functional, an increase in plasma sodium concentration is compensated by increased water intake, and number 2, by increased plasma ADH secretion, which dilutes the extracellular fluid back towards normal. So as I mentioned a couple of slides earlier, that under extreme conditions caused by complete loss of aldosterone secretion, which may be due to adrenalectomy or Addison's disease, there is increased loss of sodium by the kidneys, which can lead to reductions in plasma sodium concentration. Therefore, under extreme situations, plasma sodium concentration may change significantly even with a functional ADH thirst mechanism, but only under extreme conditions. So, therefore, the ADH thirst mechanism is the most powerful feedback system in the body for controlling ECF osmolarity and sodium concentration under normal conditions. Salt appetite mechanism is another mechanism for controlling extracellular fluid, sodium concentration and volume. Maintenance of normal ECF volume and sodium concentration requires a balance between sodium excretion and sodium intake. In general, the primary stimuli that increase salt appetite are those associated with sodium deficit and decreased blood volume or pressure. The neuronal mechanisms for salt appetite is analogous to that of the thirst mechanism. Circulatory reflexes caused by low blood pressure or decreased blood volume affect both thirst and the salt appetite at the same time. I stop here to end today's lecture at this point.